Hello, everyone. In this Zoom session, we're going to be talking about some of the fundamentals of population genetics. In other words, how do changes in individual genetic changes end up affecting and transforming a population over time? All right, so let me do a slide, sort of a PowerPoint share with you. This PowerPoint is, of course, available, on the website with sound at this point. So you can download that if you like. All right. So when we talk about, population genetics, we're really talking about, change of allele frequencies in a population. And allele frequencies, of course, as you know, are, the parts of your genome which you inherit from either mother or father, and those come together to create you. And those alleles will change over time through mutations, creating new alleles which move into populations. And if they are better alleles that produce better fitness for the population, the old alleles which were there will move out of the population because they will not be successful. Through differential reproductive success through natural selection, they will be removed from the group as new alleles arrive. So what we're looking at is a system in which we're tracking the influx of new alleles and measuring their success. And in which groups and in which areas are they successful in their adaptations. Then we're looking at how old alleles will actually either be retained because there's something good about them in particular contexts that humans need, or they're eliminated from populations because they no longer serve a useful function or are not as good as the new alleles moving in, which will, of course, give a population a competitive advantage. Now, to do this correctly, we need to start out with a really good definition of evolution. And here's one that's extremely good. So in terms of population genetics, and in an overall good definition, evolution is defined as the change in allele frequency, in the frequency of an allele in a population over time. Once again, change in the frequency of an allele in a population over time. So this simple diagram here shows, several generations of rabbits. So the first generation, we can see, there is two alleles that are present within this breeding group. And these are the relative population, like a sample population of what their alleles would be. So, we see a homozygous dominant, we see some heterozygous in there, and we see some homozygous recessive. Well, after the reproduction in the second generation, right, we can see what is the favorability, survivability of the recessive allele. Well, this recessive allele is beginning to become marginalized. We don't see it as expressed as much, but at least the homozygous recessive condition has reduced itself. And additionally, so has the heterozygous condition. So therefore, there is something far more beneficial about the homozygous dominant allele arrangement. Now look at the third generation. We can see this come to sort of fruition here where there is nothing remaining in the third generation except homozygous dominant. So we have solved the elimination of the recessive allele altogether by 3. Now, this, of course, this is a very rapid sort of change. It's meant to be that way. It doesn't happen usually that quickly whatsoever. But yeah, I hope you get the gist that we're looking at the elimination of alleles. Now, what can also happen is that we can have new alleles moving in the population. That no longer can it just be a new allele can actually replace an A variety and become dominant, and then place the old dominant into a recessive condition. And when that happens, we're starting to get allele replacements. The real idea of change in alleles, not just the frequency of alleles, but new alleles coming in. And that will be the definition of evolution. What we have to be careful of, though, is talking about recessive conditions being bad. It depends on the context in which an organism finds itself. So, for example, let's talk about our system of eye color, right, which is kind of complex, but we boil it down to in generalities. We can say that genes for brown eyes, alleles for brown eyes, are dominant over genes for blue eyes, alleles for blue eyes. And we can see that in the populations and the proportions of our populations. A far number of people have brown eyes and blue eyes. So are blue eyes going away? Is it over for us? Blue-eyed people have blue-eyed. No, not whatsoever. It depends on where the organism finds itself. Now, for brown-eyed folks and for blue-eyed folks, it depends where your population evolved in situ and what the adaptation was necessary for human groups at that time. The majority of people live at lower latitudes. This is where more of the food is, right? I mean, if you go to higher latitudes, the less the growing season, but also when you're in lower latitudes, the higher the solar radiation content is. So you need more protection from, glare light so you can see better. Remember, eyesight is one of our fundamental ways in which we negotiate our world and our survivability. Therefore, at low light levels, where the majority of people live at, we have this dominant brown, allele. Now, a smaller proportion of humans live above, the high lower light zones, where blue eyes, the ability to actually take in more light becomes more beneficial, and we can actually see better, at high latitudes. So that recessive allele represents, that small population, but a necessary population, which is why that allele hasn't gone anywhere, at least yet, that it's still present with us because humans will always be living or have been living at high latitudes. Therefore, it is necessary within a population, but at a lower percentage. Now, the thing about it is because it is a 2 allele system, not a 10 allele system, but it started as a 2 allele system. We've inherited that, through all evolutionary time. We can't exactly refine, what we mean by, 90% of human beings need just that allele and then 10% need just that allele. That's not the way nature works. We're on a system of 1 and 4 or 50-50, right? Because if you're heterozygous, 50% of the population will receive that plus the dominant, right? So heterozygous plus dominant, that'd be 75% total will receive something like the brown-eyed, allele variety, and 25%, possibly will receive the recessive condition, which is the blue eye. But if only 30% of the population, needed to be blue-eyed, we can't do that simply because we only have that particular set genetic model with percentages, right? There aren't enough alleles. If there's anything, there's a problem with the system. There aren't enough alleles in it, but we're stuck with the evolutionary descendancy of a 2 allele system. Okay, all right. So I hope that makes sense to you. That it all depends on the context of the individual to figure out if the recessive allele is beneficial or detrimental to the population. Sometimes proportions reflect their proportional need depending on what part of the population has lived to where that allele will be valuable enough. Right? I hope that comes clear in our last example of this particular slideshow, which will be on sickle cell anemia. So the definitions we have so far, which we'll need, and I'm sure you had this in the last section, of course, is recessive, which I've discussed, right? And the idea of recessivity is simply this: it is masked by dominant genes. It's masked by dominant genes for the ability, because dominant genes tend to be more valuable in a population, tend to be for a larger proportion of the group. But it does not mean recessive genes are not valuable whatsoever, and we have to be very careful how we evaluate recessive. Recessive doesn't necessarily mean to be negative. Okay. And then a new sort of definition here, which you've come across before, but I really want to reiterate it because Mendel himself did not investigate something called codominance. Codominance is when both alleles will express. Both will express, and they'll be different. They're going to be different alleles, but they both express to combine a unique phenotype, right? So the phenotype is a combination of both alleles expressed together. You can see this when you do something like breed a white rose with a red rose. What you're going to get are pink flowers. That's a hybrid. That's a combination of the two. Now, Mendel was breeding pea plants because they had discrete traits. Discrete meaning that the system of strict dominance and recessivity. Only the dominant genes would express themselves unless they were in a homozygous recessive condition. And he was tracking the appearance of that mathematically. And he realized that codominance did work. It was a system, but it was really difficult to measure that, and it would mess up his frequencies. He was trying to get at the very simplest parts of the rules for genetics. So when we talk about Mendelian genetics, we're talking about some very simple principles of genetics. And codominance has a different complexity, but it's very important because codominance works in a very similar way as population genetics works in the ideas of dominance and recessivity, where new dominant genes come in, recessive tend to move out of the back until another dominant strain will come in, and then the former dominant allele moves to the recessive condition, and so on and so forth. Well, codominance works the same way too. Is that new combinations of dominance, can come in, new alleles can replace old codominant pair, and one can leave the pair on either side. So we can see the same sort of influx of new alleles and influx of old alleles, even in dominant situations. As they affect individuals, they all want to reproduce the transformed populations. Now, with the ABO blood system, it's a great way to sort of look at our codominance. So the blood system which we have in our body is a system of codominance, but it also has an underlying, dominant and recessivity network with it too. So it works sort of a hybrid network, which is very, very common, with genetic things that happen within your body. So this is a way we can see how they will sort of work together, and some other principles here. All right. So notice that, human beings have 4 different blood types, O, B, A, and AB. AB reflects the codominant. In other words, the 2 alleles, A and B, come together. They both express, never masked, and you get a type AB. With type A and B, both A and B are dominant. So when we have AA, that's heterozygous dominant, we're going to get type A phenotype. BB, of course, we'll get type B phenotype. And the only way in which we can get O, because O is recessive, is an OO, right? That's homozygous recessive, producing type O. And then the heterozygous are operative here because O is dominant to A and to B. Therefore, we have 6 different ways of getting 4 different phenotypes, which shows us that phenotype and genotype don't always remain the same. So if I see someone's phenotype, there may be different ways of getting that phenotype that have to be investigated too, which is why when we're tracking population genetics looking at phenotypes, sometimes the phenotype masks some other things that might be happening in the real genetics of the population, because the phenotypes don't always reveal all the genotypes which are available. So why, population geneticists study genes now, because they can be more accurately tracing the transformation of a population than simply looking at the phenotypes themselves. What's also interesting about, the blood system, it shows you how one system works to be dominant over the other, and how the body can actually see these things. Is in your the AB sort of groups represent the surface proteins on the top of the cell, the type of surface proteins, which your body can actually accept as normal or reject. Thus, if you have something like an AB blood type, right, the only type of donor that you can receive, from an AB donor, would be O, right, because it's recessive, it doesn't see it. You can receive, A, because it already have the A antigen there, the body would see A as normal. You could also receive, a B, because it already has the B, and the body would see that as normal. It wouldn't be, recessive or should express. And of course, it can take AB, its own sort of blood type, right? So all of those blood groups can work together. So if you have AB blood, you're sort of blessed because, you can take, blood from all your groups. Now, it begins to diminish when we get down to the B groups, because a B can take B, but it can't take AB because it would express the positive surface antigen A, which would be seen by the cell as something negative, and bam, your immune system would tackle it and you'd have an immune reaction that may kill you, right? So Bs can only take Bs, except they can also take Os because O's recessive and not seen, right? It's masked. Well, the same thing with A. If you are recipient, you can take A blood, of course. You can take A blood, but you cannot take B, nor can you take AB because the B antigen will be there on the surface of the blood cell and your body will attack it. But you can take O, because O would be invisible to the system because it doesn't recognize it. Essentially, it's shut out. That's what we mean by recessive. So when I look at O, something really positive comes out with this. If you are O, type O, which means you're homozygous recessive, you can donate to anyone within the group. You are a universal donor. The particular problem is that being a universal donor also means the only people that can donate to you are those with OO, right? The actual homozygous recessive condition. So there's a negative to that also, right? And this system is always a system of positives and negatives. It depends on how you look at it and whether you're the donor or the recipient. Okay, move on. Here's a great example of genetics in action in populations. when it comes to trying to evaluate recessive versus dominant conditions and mutations and what they can do to a population. So we'll spend a little time with this. You're looking at a molecule in your body, a macromolecule called hemoglobin. This is in all your red blood cells, and there's lots of it. And this macromolecule, which is consistent of 4 proteins. 2 of them are exactly the same. So there's 2 sets of proteins, right? You have 2 beta proteins and 2 alpha proteins. 2 beta, 2 alpha, and they come together. And each one of these protein chains has a little heme group, a little iron group in the center. And that job of that is to pick up oxygen in your lungs and deliver it to your cell. That's how you get oxygen into your body. Okay, so what hemoglobin does is this macromolecule. Okay, so this hemoglobin B molecule is coded. The hemoglobin molecules are coded on genes, and one particular the chain here, the beta chain. You see the beta chain, you see the alpha chain, you see the beta chains. The beta chains are coded on chromosome 11. Now, here's what's interesting about this arrangement here, is that you see there's 2 beta chains. Well, one beta chain comes from the allele from your mom, and the other beta chain comes from the allele from your father. Okay, so they both come together and they're both expressed. All right, so they're both expressed. So beta chain from mom, beta chain from dad are expressed. But this also means that you can get changes on the beta chain from dad or the beta chains from mom. So you can get independent mutations, which can either mess up, one allele or one protein, or both proteins, depending if you have mutations in both mom and dad, and with differential disastrous effects on this entire molecule. Let's look at what can happen. So let's say that we're on chromosome 11 and we're coding, this is a protein coding sequence on a gene for the beta molecule, the beta, right? The beta, molecules. There's 2 alphas on this side, and there's 2 betas. All 4 of them carry oxygen, 2 betas, okay? So this is on chromosome 11, and what might happen is, if you can see, you see the sequence of normal cells in the top, and look for a protein called glutamine at the very end where it says CTT. What can happen is that CTT has experienced a point mutation, and it's changed that T to an A. So when you change the T to an A, it changes the amino acid from glutamine to valine. Now that is going to have a dramatic effect on that protein when it actually goes through transcription and translation. Now, if we do this, let's have a look at the effect on the cells. Voila. We have essentially mutated one of the beta chains. But here's the thing, in this situation, what we're doing is, what I haven't told you guys is that to get a condition where that kind of like that sickly looking orange cell is there, we actually have 2 mutations that have happened. One in mom's allele and one in dad's allele in the same condition. They both have had that same situation. They both have had an allele which was mutated. They both have given the progeny, a new allele, which is a recessive condition. And when they come together, the proteins, the beta chains don't work. They don't hold oxygen. They don't pick up oxygen. And they also cause the red blood cell to fold down. And when they fold down like that, you get a condition called sickle cell anemia. There it is. Sickle cell anemia. That's in the homozygous condition, which we call HB, which that's the hemoglobin, the beta chain S, and the S would be abnormal. So you can see it occurs in an individual homozygous recessive. HB, hemoglobin B, S was abnormal. So 2 individuals there, mom and dad, are both carrying this allele, which is the S. Okay, the A would be the normal condition. The S is the abnormal condition. Okay, so these things don't carry oxygen very well, if at all, and they fold down like that, and they can glump together. They can cause serious blood clots. And later on in life, by your teenagers or your 20s, you may experience death. It's just generally a life killer. It's a terrible, terrible disease. But here's the thing. If you're lucky enough to have a parent that only gives you one mutated, beta chain. In other words, your mom has the mutated, version of the HBS, but dad is HBA normal in the heterozygous condition. That means you're going to have a red blood cell that only one chain is a dud. And that means that red blood cell is only going to partially fold up. It won't look sickly like that banana-shaped little poor sickle cell anemia cell. It'll look a little bit different. It'll look a little bit different. It'll still carry some oxygen. You may feel a little bit not as much endurance. You might not be as great and sprightly as all the other runners out there in the world or whatever you do, stair climbers and stuff. But you'll live. You'll live. So it's sort of a progressive thing, depending on if you have the full-blown recessive condition or if you have the heterozygous condition, okay? Now, on surface, this looks like a negative thing, like, oh, that allele's in the population, when's it going to go away? Well, it hasn't gone away. In fact, it's still there at a significantly high proportion. In evolution, negative alleles that affect the population negatively always go away. So why is this hanging around? We got to investigate this. When we look at a Punnett square, we have to compare this to the sickle cell disease. And then we have to compare this to where individuals actually live at. So the A here is, of course, the normal variant, and the S is the abnormal variant. And remember, each parent on each side carries, a gene for a different beta chain. They're going to produce their own individual beta chain, which come together to produce the macromolecule. So if we have individual parents who are carriers, right, they're heterozygous, they carry a normal chain, they carry an abnormal chain. When they come together, a typical Mendelian mating will occur, and we'll have one normal, completely normal, two normal chains, heterozygous dominant. We will have two heterozygous individuals, that's A with the S, and normal with the abnormal. And of course, on average, we'll have 25% of the population, which will have a homozygous recessive condition marked by the SS. Okay? Well, so what does this do for a population? I mean, how does this transform populations, or how does this benefit populations? Believe me, if a allele is there, it's there for a reason. And the sickle cell trait, the SS trait, is there for a reason. If this individual or a group of individuals live in regions of high malaria, endemic malaria, this can be highly beneficial to our species and our populations, and eventually to our species. For example, the cell in which you are looking at back here, these red blood cells, right? A normal healthy cell, which you see on the outside there, can be easily penetrated with pulsopar plasmodium. Just think about it as malarial parasite. It's a single cell parasite. They can enter your red blood cell, and it will eat and digest the interior parts of your red blood cell and grow and grow and grow until it explodes your red blood cells and move to the next red cell, and they're hemophagic sort of things blowing red blood cells up, and you can eventually become extremely sick or die of malaria. Okay? Now, if your cell sickles, if it actually has an S variety, and it sickles down, and the plasmodium, the malaria parasite, cannot enter the red blood cell. Now, here's the thing. It can't even enter the red blood cell if you just inherit one recessive allele. So if you have the heterozygous condition AS, that's enough to cause a partial conformational shift of that red blood cell. That plasmodium, that falciparum, that malaria will not get to you, will not kill you. If you're in a heterozygous condition, you're still going to live. You're not going to have as much endurance as other humans, but it's not going to affect you other than that. Okay. So in a condition of high malaria in a population, you're the only one that's going to survive if you're heterozygous. What I mean, if you have no protection, if you have two normal beta chains, that means that cell is going to get infected by malaria. You're probably going to die. But on the other hand, if you end up with the recessive condition, fully recessive condition, homozygous recessive, unfortunately, you're going to die of sickle cell anemia. So what's the ratio of death to life here? 50% will live with the heterozygous condition, and 50% will die. Like, oh my God, 50% die? Yes. But think about this. If there was no recessive condition, how many people would die if everybody was normal in the presence of malaria? Everyone would die. Because we only have a system of 2 alleles, that's the best our system can do to protect our population. The genetics and the phenotype of the population is this. The best we can do is 50% will live while 50% will die. And we can consider this, the heterozygous, probably advantage here, with some discrepancies that 50% of the population will go to the wayside. But when we're talking about population genetics and evolution in general, we're not concerned about individuals. We ourselves might be. I'm concerned about my life and yours too, because I love everybody. But that's not the way that it works in evolution. Evolution is about the population. How do you best preserve a population? And this heterozygous example completely does that, which gives us a lesson to why sometimes we do see recessive, traits like this S variety that causes that conformational change in that beta, which seems like such a negative thing, can actually be a positive thing if the context is correct, just like folks like us with blue eyes. All right. Now, there is a film, Malaria and Sickle Cell Anemia, that I do want you to watch. So the link to this is right below this. You can click on that, Malaria and Sickle Cell Anemia. It talks about a man named Tony Allen who went to Kenya to try to understand the genetic mechanisms behind this, how this actually works in the context in which this becomes very protective. It is a great story, and I want you to watch it. Okay, you can have a question of that on your next quiz, I am quite sure. Okay, then you can finish with this. This is just a little wrap up of what we talked about this in the film. If you've gathered what I've said, so far about the protective qualities of the recessive alleles and the heterozygous condition, understand that from the film. You don't have to revisit this last show, but if you want sort of a capstone with it, this little last slide, we'll provide that for you. All right, so I'm done for the day. I'll see you guys in the next Zoom lecture.